Hello, and welcome to the second installment of the 2016 INSAR Summer Institute. I'm Carolyn McCormick, co-chair of the INSAR Student and Trainee Committee and a postdoc at Brown University. The INSAR Summer Institute is a 6-course online series presented by the International Society for Autism Research. The goal of the Summer Institute is to provide a forum for the presentation and discussion of autism-related research topics that is accessible for people new to the field. The Summer Institute was originally motivated by and conceptualized for early career researchers, specifically graduate students and postdocs, but is open to all those interested in autism research. We hope that the Summer Institute provides an opportunity for scientists with different backgrounds and from around the world to learn from each other and ultimately advance our understanding of autism. The Summer Institute sessions are free for everyone to attend live and available to INSAR members for replay through their My INSAR account. This is the second year we've offered this training opportunity, and the theme for the presentations is familial aspects of autism. Today is the second session in our series and features a presentation from Dr. Joseph Piven, who will be talking about infant siblings of children with autism. Note that the background materials, there are background materials for today's talk. If you haven't already, you can download these now at autism-insar.org. These materials define some of the key terms and provide suggested readings in the literature. As with all of the Summer Institute sessions, a group of trainees worked with Dr. Piven to prepare these course materials. The working group was led by Ashley Stevens, doctoral candidate in educational psychology at the University of Utah, working with Stacy Shumway-Manwaring. Megan Swanson and Robert Emerson, postdocs at UNC, working with today's speaker, Joe Piven, also contributed to the working group, and Dr. Megan Swanson will be monitoring today's discussion. Before we get started, know that you can ask questions at any time. We will leave plenty of time for discussion during the back half of the session. Ask a question at any time using the chat window on the left of your screen, and we'll get to as many of them as possible during the discussion period. At the very end of the session, we will have a few minutes to talk about career development. If you want to get some advice about career development topics from Dr. Piven, please post your questions in the chat window and we will incorporate them in our discussion. We also greatly appreciate feedback from attendees. When you end your session, please take a few moments to tell us about your experience today in the comments section. Now I would like to introduce Dr. Joseph Piven. Dr. Piven is a child and adolescent psychiatrist specializing in the clinical care of individuals with autism and other neurodevelopmental disorders. He received his MD from the University of Maryland and completed training in the genetics of neurobehavioral disorders during a postdoctoral Merck Fellowship at Johns Hopkins. He was at the University of Iowa for several years before joining faculty at the University of North Carolina at Chapel Hill, where he is the Thomas E. Castelloe Distinguished Professor of Psychiatry, Pediatrics, and Psychology, and the Director of the Carolina Institute for Developmental Disabilities, CIDD. The CIDD is a comprehensive institute for services, research, and training relevant to neurodevelopmental disorders. His research has emphasized interdisciplinary collaborations in brain imaging, behavioral, family, and molecular genetic studies aimed at elucidating the pathogenesis of autism. He directs a T32 postdoctoral training program in neurodevelopmental disorders and is director of an NIH-funded Autism Center of Excellence Network study of brain development in infants at risk for autism. He is the author of over 150 peer-reviewed publications on neuropsychological mechanisms, brain morphology, and the genetics of autism and Fragile X syndrome. He's the founding editor-in-chief of the Journal of Neurodevelopmental Disorders, the aim of which is to promote interdisciplinary research on the pathogenesis of a range of neurodevelopmental disorders. We'll now turn it over to Dr. Piven for his talk entitled Infant Brain and Behavior Development in Autism, Relevance to Intervention and Pathogenesis. Thank you for joining us, and welcome, Dr. Piven.
Okay, well, thank you, Carolyn. Thank you for that lovely introduction. And I wanted to thank several of the people that you mentioned. Megan Swanson and Robert Emerson have been critical for my ability to actually do this because I am kind of computer illiterate. Ashley Stevens, Carolyn McCormick, and Tessa O'Sullivan. So go to the first slide. So this is a talk about babies. It's a talk about babies at high familial risk for autism and it's a talk about development of brain and behavior during this period of time from infancy through toddlerhood, when we see the unfolding of autistic behaviors and brain-related abnormalities. What I'm going to do is mostly talk about the work from our infant brain imaging study. That's an Autism Center of Excellence network. I'm going to present a fair amount of unpublished work, and I'm going to try and, at least at the beginning, tell a little bit of the story of how I think that this work unfolded. Okay. Well, the story really begins with this slide about familial risk for autism, and this is a paper by the Baby SIBS Research Consortium of Autism Speaks, with Sally Osinoff as the first author. And it's probably well known to most of you, but once you have a child with autism, the risk of having a subsequent child with autism is 20%, around 20%. And so that's quite a bit greater than what we see in the general population. And that enabled us to move from finding very young kids, which we've done at, say, 2 years of age, that are referred through the community, to basically an unbiased, with respect to later diagnosis, sample of infants, and to be able to watch them as they develop. Okay. This work is a kind of a caricature of a table or figure in work by Lonnie Swagenbaum and Susan Bryson that really, I think, was the first in the field to kind of kick off this research. And it basically uses the Autism Observation Scale for infants. So it's a kind of a syndromic approach to thinking about the various aspects that may go into a definition of autism in infants. And you can see on the right that it has social features and also non-social features. And what was, I think, the excitement at first about this study is that we had moved from being able to identify children with autism in the preschool period to toddlerhood. But what this study showed was that we could actually see differences in kids that went on to develop autism, that's the red line, from those high-risk kids that didn't develop autism, the green line, by 12 months of age. But what wasn't entirely clear and discussed, at least in terms of what I understood, was what was happening at 6 months of age. I can get this pointer. Yeah, at 6 months of age. And you know, when I first saw these data, I wondered, well, is this evidence that there are no differences as early as 6 months of age in these behaviors, or is this just an artifact of kids with very limited behavior repertoires and our inability to really measure it? But here you can see another paper by Sally Osinoff that really, I think, makes the point very clearly. These are experimental measures of various kind of social behaviors, and she's comparing a group of kids, high-risk kids who develop autism, the line in red, to a typically developing contrast group. And here she shows very clearly that there's a period of overlap early on in the first year, at 6 months, where these kids don't really look any different on these social measures. But as you can see, these social deficits emerge in the latter part of the first year and in the second year, and out to 36 months of age. So I wanted to show a few videos. I thought it might be make it a little bit more interesting, but really to illustrate this point of what do kids that are 6 months of age look like. And see if I can get this started. Oh, this is the wrong one. Click the play button. All right. All right. Oh, no, that's not the right one. Oh, yeah. Okay.
That's silly.
I have no sound. I don't know if other people have sound. But what I want to show you here is that this is a typically developing 6-month-old, the daughter of our project coordinator. And as you can see, she was a very social little being. She was playing peekaboo with her mother and reacting, showing reciprocal affective responses in a very normal way. Now this is a video that I got courtesy of Lonnie Swagenbaum and his Canadian group of researchers. And it's of a young boy who has an older sibling with autism. And as you can see in this first, whoa. Good. All right. Now, I'm not sure if you guys have sound or not. I can't hear anything, but it's okay without sound. So this is a little boy who is about to play peekaboo, and if you could hear him, you'd see that he was responding vocally and certainly is engaged socially in a very typical way. This is just a snippet that you have to accept that it generalizes to the rest of the video, but there's no response. It's a very different response at 12 months of age in this little boy. He was just not having the same kind of social response. Now this, unfortunately, is not a segment that reveals very much because we really can't see his face. But what I want to do is tell you about the next segment. And this is with a little boy sitting on the lap of his parents. And Lonnie Swagenbaum, if you could hear him, he's literally yelling across the table this little boy's name to try and get him to respond. And what you can see is that he's not at all interested in Lonnie or his parents. He's really preoccupied with manipulating these objects that are in front of him. And so that's a very clear difference in progression over time, particularly of these social behaviors. So sort of in parallel with this, there's been development of some early findings in brain imaging, and we got interested in this early in the 1990s, thinking about brain enlargement. And we reported in a couple of papers brain enlargement in adolescents and adults, and we replicated that. And the findings sort of lay dormant until the early 2000s when Eric Orshane published a paper showing very robust differences in young kids at 2, 3, and 4, suggesting that the differences were even more robust at that age, which is something I think that the field has pretty much confirmed. So we conducted, with Heather Hazlett and our group at UNC, a longitudinal study of 2-year-olds, and these are 2-year-olds not at high risk, but 2-year-olds that we identified in the community with autism, so a more severe sample. Red is the autism group, blue is our control group. And what we saw was that we found enlargement in brain volume as early as 2 years of age, and it was maintained throughout the whole interval of our study until age 4. The slopes were no different, suggesting that it is a period before age 2 when this difference came about, this brain overgrowth. And then if you look over to the left of your slide, we did one of the early sort of head circumference studies, and we had a local group of kids that were systematically diagnosed, and we compared them to controls who were comparable on age and SES and gender and several other things. And what we found was that these two lines, the red line is the kids with autism, and this is, by the way, retrospective head circumference data from the medical record. And what we found was that there were no differences in the trajectories of head circumference in the first year. The differences really began to emerge after 12 months of age, suggesting that there was something going on with the brain enlargement in a very simplistic way that was increasing head circumference, and the suggestion was that it was happening in the latter part of the first year. So when you juxtapose these findings with those by Lonnie Swagenbaum or Sally Osinoff, what it suggests is that there is a sort of a striking overlap of changes in the behavior in kids who go on to develop autism in the first year, between 6 and 12 months of age. And this evidence of brain overgrowth around the same period of time, at least prior to age 2, and perhaps beginning at the end of the first year of life. So this is what prompted us 9 years ago to begin this study, this Autism Center of Excellence study that we refer to as IBIS, or the Infant Brain Imaging Study. And the overarching sort of goal of this was to find as many high-risk, high familial risk for autism infants. So those are infants who have an older sibling with autism, and low-risk controls, so they don't have first or second-degree relatives with autism, and to examine them at 6, 12, and 24 months with brain imaging and behavioral assessments. And as you might imagine, this takes a lot of sites and geographic regions to generate enough of a sample to conduct a study like this. And so when we started, we had 4 data collection sites that we still have at Children's Hospital Philadelphia, UNC, WashU in St. Louis, and University of Washington in Seattle. And as you can see, we have a number of other folks around the country that are critical to supporting these efforts as well. And so now I want to try and start talking about some of the data from this study. And this is a published paper by Annette Estes and Lonnie Swagenbaum and the rest of our group. And basically it presents behavioral data in our high-risk group, which at the time was 274 subjects, versus our low-risk group of 105. Now, we do a clinical best estimate diagnosis at 24 months of age based on DSM-4 that includes information from ADOS, ADI, and Mullen, and any other pertinent information that we have. And in this sample, we identified 49 that met criteria for DSM-4 autism. And then, and 215 in green who were what we call high-risk negative for autism. And among those with autism, we subset the group based on ADOS scores into a more severe and a more moderate group, as you can see, 38 and 21. So we went back to the AOC that I've already talked about, and what we found at 6 months of age was just like the paper by Lonnie Swagenbaum in 2005. We found no differences between our low-risk negative, our high-risk negative, and our 2 autism severity groups. But by 12 months of age, those differences had emerged. So there were significant differences between our sort of autism, eventually autism negative group, diagnosed at 24 months of age, versus 12 months of age in those kids that went on to have a diagnosis of autism. So I want to just show you a little bit of data from the study, and here we have the Mullen test of cognitive development, and for those of you not familiar, this is done by an expert tester, whereas the Vineland Adaptive Behavior measure is parental report. So 2 sort of distinct ways to collect these data, but pretty much the same result. So over here on the left, this is the Mullen composite. We see at 6 months of age, no differences, but the differences emerge by 12 months of age, and there is a significant effect over time with showing the symptoms unfolding over time, so that by 24 months of age, we see much more severe cognitive deficits in the group that went on to have autism, less so in those that had moderate autism than those that had more severe autism, and pretty much identical results on the Vineland. So I just want to kind of drill down a little bit on some of these data because obviously there are some more fine-grained wrinkles in what we found. And the first is here, looking at the Mullen gross motor development. Now, this was a finding that was first reported by Becky Landa a number of years ago, and that is that infants that went on to have autism had these gross motor deficits, and we didn't have good measures at the time in this study, but as an example, when you pull a 6-month-old to a sitting position, they should keep their head erect, and if their head lags, then that's evidence of immature sort of gross motor development. So here what we saw was in those kids that had, we saw significant differences as early as 6 months of age. And in that kids with autism had lower gross motor development than the kids without autism. Here, what was interesting to us was that those differences did not change over time. They seemed fairly stable. And contrast that with what we saw on the visual reception subscale of the Mullen. Now, I'm not a psychologist, and many of you will know much more about this than I do, but visual reception is all about sort of tracking in the environment salient information. So very much visual sensory index. And here we saw significant differences as early as 6 months. But these differences really unfolded over time. Now this is kind of a third variation on the theme, and it's really meant to show you some evidence of the sense that we had when we saw these children of real variability during this interval, evidence of plasticity in their brain and behavioral development. And what we see here is on the Mullen fine motor scale, what I want to show you is this group, this sort of brown line that develops moderate autism symptoms, what we call spectrum. And they're no different from the rest of the group at 6 months of age, but they are different from the autism group at 12 months of age, and they're really sort of right in the middle of the high-risk negatives and the low-risk negatives. And then by 24 months of age, they take a turn to looking much more like the kids with more severe autism. And if I were to show you individual trajectories, you would see the same thing. We see children who start looking like they are headed towards autism early on, but then later on kind of veer to more typical development and vice versa. So this is a time of, I think, great change in behavior. Now here's one other study that took a different approach from our group. Jed Elison was a graduate student at UNC at the time, and he's now on the faculty in child development at University of Minnesota. And Jed was interested in this gap overlap paradigm where we used eye tracking and first showed a stimulus in the middle of the screen. For this condition that I'll tell you about, the overlap condition, we left that stimulus on and added a peripheral stimulus. And this is sort of a measure of visual orienting to salient information, as I've written here in the environment during infancy is critical for early cognitive development. And the aim here is to see how quickly infants shift or orient their gaze from central to peripheral stimulus. And here's some of the data. And the bottom line was that we saw slower orienting in the kids that went on to develop autism, the high-risk positive kids. They were slower than the high-risk negative kids who were no different from the low-risk kids. Now this is a finding that while the data are a little different and at different ages, Mayada Elsabah has shown, and others have studied this as well. So we think this is a real phenomenon. There have been some explanations about this idea of problems with attention disengagement, or what people have talked about as sticky attention, so that kids get kind of, with autism or sort of prodromal autism, get stuck on the initial stimulus and have difficulty moving to the second stimulus. So I just want to summarize where we are now with the findings that I've presented. There are converging findings from our behavioral studies and from the behavioral studies in the field. And they point to a prodromal or pre-symptomatic, at least for the defining features of autism, period in the first year of life in the children that go on to have autism, where they have motor deficits and they have sensory deficits and deficits in visual orienting or attention, but they do not have the defining features of autism, and in particular the social deficits. The social deficits, on the other hand, unfold during the latter part of the first year and the second year, and that's accompanied by declining cognitive abilities and adaptive function across that interval from 6 to 24 months of age. And then, as I mentioned earlier, the finding with fine motor development and other areas as well. Well, when you look at on the individual level, there's tremendous variability of trajectories across this period of time. So this is a slide that's supposed to impress you because the image on the right is supposed to be pulsating between the size of the neonatal brain at 2 weeks and the size of the 1-year brain. But I understand that we can't do those kind of theatrics in this presentation, and so I'm just going to refer you to this very dull bar graph here, where you see that the size of the brain, the total brain volume of a 2-week-old, really doubles by the time this child gets to be 1 year of age. And this is in a typically developing population. Then when you look over here, you see that doubling, but really by 1 year of age, and certainly by 2 years of age, the brain volume in kids is pretty much like that of older kids or adults. And this is a pretty crude way to kind of illustrate the point I want to make, that there's a tremendous amount of variation, certainly during the first year and the first and the second year of life in typical development and in aberrant development as well. So what we aim to do with the brain imaging part of this study was to look at these three questions. What are the earliest postnatal brain manifestations of autism? What are the developmental trajectories or the sequence of brain metrics as they develop over time? And what are the brain behavior relationships longitudinally? So here I want to show you begin to show you some of our data, our unpublished data. And we think to some extent we've been able to answer the question here of when does this brain volume overgrowth happen. And what you see here are kids, high-risk kids that develop autism in red, high-risk kids that don't develop autism in green, and high-risk and low-risk kids in blue. And these are kids that have brain imaging data at all three time points. And what we're able to see here is that there's really not much difference in this slope of brain volume development in the first year between 6 and 12 months. But between 12 and 24 months, the growth rate increases significantly. And when we look at differences at 12 months, we don't see significant differences, whereas we do see significant differences at 24 months. And so the conclusion from this is that there is early postnatal brain overgrowth in autism, and it seems to be happening, at least in this sample, in the second year of life. And I probably should qualify that by saying that this sample is probably different from a community ascertained sample for several reasons. One, that they tend to be much milder because we ascertain them in a different way, and we sort of have pretty rigorous surveillance during their development. And also the fact that they have a different sort of genetic architecture. They have generally are thought to have familial autism. So we took this observation about increased brain volume, and we did something that's become fairly popular in the field to try and disaggregate the parts of volume increase. And we looked at both cortical thickness and surface area. So if you have a rectangle, this is basically the height of the rectangle, or here, these red lines, cortical thickness, and the kind of sulcal gyral ins and outs of the surface of the brain, which is the surface area representation here. And surface area expansion is kind of a critical part of human development. It's what really distinguishes mammals and humans and allowed us to have larger brains. But several things that are interesting about this, other than the fact that we can measure this, which is not a simple thing to do at 6 and 12 and 24 months. The off-the-shelf methods don't work. But there's a large literature on surface area and cortical thickness in humans and animals showing that the genetic underpinnings of these are different. So surface area genetics is not the same as the genetics that's underlying cortical thickness changes. The second is that there's a literature, particularly in animals. We can't really get this data very well in humans, but it shows that changes in surface area or increased surface area is related to symmetrical proliferation of sort of baby neurons or neural progenitor cells. So it links us to a sort of a pathophysiology on the cellular level. And then the third is that we actually reported this several years ago in our sample of 2 to 4-year-olds with autism, where we showed increased surface area but not cortical thickness that was present at 2 years of age and maintained across the 2 to 4 year age interval. Okay, so that's the background for surface area, and we're going to have to move back through these slides again. That's the background on surface area. And here are the data. So again, autism in red, high risk who don't develop autism, or high risk negative in green, and the low risk group in blue. And what we see here is a little different than what we saw in the brain volume measures. Here we see that there's an increased rate of expansion of surface area in the first year, in the period between 6 and 12 months of age, such that these children who go on to develop autism don't look any different at 6 months of age, but they are significantly different at 12 months of age, and that difference is maintained with no increase in growth rate during the second year. So we have increased surface area growth rate in autism from 6 to 12 months of age, and I wanted to add no differences in cortical thickness. So we naturally looked at where this was happening most robustly in the brain, and we were able to divide the brain into a large number of what's called regions of interest. And the red spots are the spots that were the most strikingly different in terms of hyperexpansion of surface area. And those are in right middle occipital cortex, right middle occipital gyrus, and left cuneus, areas that are in the visual cortex and visual sensory regions. So I mentioned that this idea of increased proliferation in surface area, but we were interested in seeing to what extent our surface area findings were related to later brain volume findings. So we looked at the correlation and found that there was a temporal relationship that that increased surface area growth rate from 6 to 12 months in autism was significantly correlated with increased brain volume in the second year. So then the next question always comes up about how does this translate into behavioral changes? And So we were interested in looking at to what extent brain volume changes are linked to the emergence of autistic behavior. And so what we did was we looked at change in brain volume in the first year and change in brain volume in the second year versus overall ADOS measure of severity of autism. And what we found was that there was a significant relationship in the second year, but not in the first year. So then we were able to subset the ADOS into the social affective score and the restricted repetitive behavior score. And it looked like the differences were largely due to differences in social affective, the social affective scale. We didn't find differences in the restricted repetitive behavior. That may be because the instrument is not as good in measuring that. We don't know. But we clearly find differences in social behavior. And we were fortunate enough to have another measure of social behavior that was not developed for autism, the Communication Social Behavior Scale, or CSBS, as an independent measure of social behavior at 24 months. And there again, we found that it was the change in brain volume in the second year that was most related to social behavior, severity of social behavior deficits, and not changes in the first year. So all this suggests that change in brain volume from 12 to 24 months is linked to unfolding of social behavior deficits. Now, this is not to say that other things aren't going to be linked as well, but there is evidence that that volume change is linked by these data. So when we take all this together, it leads to this story that in the first year of life in kids that go on to have autism, or at least in high familial risk infants, they don't look like they have the defining features of autism, particularly at 6 months of age. But they have sensory, visual sensory and motor problems, and visual orienting or attention deficits. And also in the first year, we see this hyperexpansion of surface area from 6 to 12 months. And that's temporally correlated with the subsequent overgrowth in brain volume in the second year. And that brain volume overgrowth seems to be related to severity of social deficits in the second year. So I just want to just shift for a moment into some of the more basic science biology that at least is consistent with this finding and suggests that this is some possible plausible mechanisms. And the first is a paper by Eric Korshane and JAMA 4 or 5 years ago that reported increased neuron number and size in prefrontal cortex in a post-mortem sample of children with autism. Then there's a series of studies that really start with this New England Journal paper by Lauren Weiss back in 2008 that identified a copy number variant, a microdeletion, and a microduplication in this area of 16p11 that seemed to be more common in people with autism. This next study by Qureshi et al. Through the Simons Foundation VIP project looked at imaging in a large sample of people with 16p11 deletions and duplications. Now, this is not necessarily folks that have autism, but clearly autism is associated with this copy number variant. And what they found was that those with the deletions seem to have brain overgrowth, whereas those with the duplications had smaller brains. Now that allows us to go into a mouse model of 16p11 deletion in autism, and this paper shows that those mice with brain overgrowth and 16p11 deletion had an increased cortical progenitor cell proliferation. And also some other abnormalities in cytoarchitecture and signaling pathways. So this is an example of a specific genetic abnormality, sort of syndromic autism, linked to brain volume that shows increased proliferation of neuronal progenitor cells. And actually, the latest paper, I believe, that came out this week shows differences in white matter fiber tracts and white matter volume in 16p11 deletion and 16p11 duplication individuals from the same group. And finally, there's a paper, I apologize for this, Journal of Neuroscience 2014. I don't know how that wandered on here, but this is a paper by Fang et al. And Cell Reports where these investigators were able to experimentally increase the number of cortical neurons, pyramidal neurons, in the upper neocortical layers by injecting a particular molecule that expanded the progenitor cell population. And what they found on this sort of classic three-chamber task from Jackie Crawley looking at social behavior in mice is that there were clear social deficits in those that had increased brain volume. So again, a link to mouse models. So now I want to shift and talk about a little bit more fine-grained level of analysis than our brain volume, and that's something called diffusion tensor imaging, or DTI. And this is really based on the fact that we can measure diffusion of water through the brain. And the sort of example to describe this is if you put a drop of ink into a glass of water, it would diffuse in all different directions. But if it's within a straw, sort of simulating a neuron, then it diffuses in a particular direction with a particular force. And this direction and force is what we measure in diffusion tensor imaging. And we get these pictures of white matter tracts, and we can extrapolate these sort of cartoons, kind of spaghetti-like images that show us particular fiber tracts, like cortical spinal tract and others, and we can measure properties of those tracts, properties of the diffusion of water, and we don't entirely know what that means or how that translates to pathophysiology, but in a very general sense, we all have seen that that it's an index of maturation, because these DTI differences show clear changes in our sample, even across a 1-month interval in the first year of life. So this is a paper from our group by Jason Wolff, also on the faculty at the University of Minnesota, where he hypothesized that 15 neural circuits based on the literature in autism and presumed underlying brain behavior relationships might be relevant in thinking about brain development and autism. And he used diffusion tensor imaging and a metric of this called fractional anisotropy to look at high-risk infants in red versus high-risk infants who developed autism versus high-risk infants who did not develop autism. We didn't have a large enough low-risk control group at the time of this publication. And this is just an example of what he found in one tract, the inferior longitudinal fasciculus. And what you'll see here is that there's significant differences between the trajectories of these tracts. These differences are present by 6 months of age, where we see an increased fractional anisotropy, or FA. They are no different at 12 months of age, but sort of reversed at 24 months of age, with FA being lower. Now, we don't know that the same mechanisms are operating at both points along this way, but we do know that these are sort of interesting, striking changes that appear very early and change over time. And looking at the full 15 tracks, 12 out of those 15 tracks were significantly different between high-risk autism and high-risk negative group. But really all of them have this same crossover pattern that you can see sort of this X that's striking in the sort of generalization across all the tracks of the same finding. So what we concluded from this was that brain changes are present in autism as early as 6 months of age. And the important point here is that this is before the appearance of the defining features of autism. So this is when, in the video by Ami Swagenbaum, the little boy was showing very good social reciprocity playing peekaboo. And the second part of this is that there are age-specific brain changes in autism from 6 to 24 months during a period of time when autism is first unfolding. So we find a number of examples of similar changes where we see differences as early as 6 months of age and change over time. And this is one of them. We've in the field measured the corpus callosum, this major white matter tract in the center of the brain going anterior to posterior. And for a long time, what's been striking in the field is that we had this finding of increased brain volume, yet all of the studies, or most of the studies, suggested that the size of the corpus callosum proportionate to that overall brain volume was actually decreased. Now in this study, also by Jason Wolff, published last year in Brain, he found significant enlargement early on in the anterior part of the corpus callosum, this dark red, and it kind of normalized as the children went from 6 to 12 to 24 months of age. So the idea being that there, again, is a difference as early as 6 months of age that shows age-specific changes. So it's not the same at all time points. In fact, if we were to look for it at each time point, we would find different results. I'm moving forward with this idea and linking brain and behavior. Jason has data that he's presented at INSAR this year with high-risk autism and high-risk negative, looking at particular pathways that might underlie ritualistic, repetitive, and sensory behavior. And he looked at a number of different pathways, and there are a couple of take-home messages here that I present really more as just examples of the kinds of things that we've found in this data set. The first is that there are changes at 6 months of age in fractional anisotropy of the corpus callosum genu and the cerebellar pathways that were measured. And these related to later repetitive behavior and sensory behavior. On the other hand, we didn't see an association between these structures and social affect score, so there's specificity of the outcome. And we didn't see the same differences in areas that we expected to find them in that are often related to more repetitive behaviors, cortical spinal tract and anterior thalamic radiation. And that's a similar theme that we found in other studies we've conducted as well. A paper last year, I believe, by Megan Swanson showed language development in a typically developing population that was related to fractional anisotropy in the splenium, but not in some of the more traditional areas or structures that would be expected to be related to language. So now I want to switch to something that's very different and literally sort of outside the brain. And this is a report of a finding by Mark Shen several years ago when he was a graduate student with David Amaral at the Mind Institute. And what they were able to show in a relatively small sample, 10 kids with autism and other out of 33 high risk, and incomplete data across the different intervals. But nevertheless, they were able to find brain enlargement and elevated what was called extra-axial fluid volume, or extra-axial CSF, in infants who went on to develop autism. So I think this is sort of a testament to the robustness of that effect. And what they particularly found was that this sort of thin rim on top of the brain in the subarachnoid space was increased, and that's where cerebral spinal fluid is. This is in a typically developing person, and this is in a person with autism who developed autism. So fortunately, in our IRIS data set, Mark has been able to replicate and expand this finding in a larger sample of 47 kids with autism out of 22 high risk, 221 high risk and 122 low risk, and found virtually the same thing, that this data showed that by 6 months of age, there was a striking enlargement in extra-axial fluid volume that persisted, perhaps more modestly, but persisted across this entire interval. And when he subset the group of kids with autism into those with more severe symptoms and less severe symptoms, he found that the differences were really pretty much due to the more severe group, where the enlargement was even more striking, increasing from 11% to 22%. Now, we have a parallel sample of infants with Fragile X syndrome that we're studying in exactly the same way as we're doing with kids at a high familial risk for autism. So as I think most of you will know by now, that Fragile X syndrome is due to this expansion of a triplet repeat in the Fragile X site in the X chromosome, and it alters something called Fragile X mental retardation 1 protein that affects the development of dendritic spines, results in the most common form, heritable form of intellectual disability, and autistic features and DSM autism in about a third of the affected individuals. So when we looked at these kids with Fragile X, that's the group in green, and what was very striking to us is that the differences were even larger at 6 months of age and persisted. And the reason why this is exciting is that for a long time, people didn't know what the CSF did, and they thought of it as just sort of an extra cushion around the brain to protect it. But what's become clear over the last several years by several labs around the country is that the CSF is really an active participant in metabolism in the brain. It delivers growth factors. It also is sort of the cleaning filtering system. It filters out metabolites, things like amyloid beta and pro-inflammatory cytokines. And there's new data in Alzheimer's that perhaps it plays a role. And so the CSF has become a much more exciting target. And the idea that there may be some early flow problems in autism in the sort of filtering of the brain that may have some downstream cascading effects, I think is very plausible and very exciting. So just to talk a little bit further about the Fragile X comparisons, I think we're very excited about this because it gives us a little idea about specificity, so we can compare kids with Fragile X versus kids with idiopathic autism, or at least unknown to us. The causes are unknown at this point. And we don't have a large enough sample of Fragile X kids in this study to subset out those that meet criteria for autism. But what we see here is a finding that we've reported with Alan Reese at Stanford in 2-year-olds, that there's a robust increase in Fragile X in the caudate nucleus volume at 24 months of age. But we now see that those differences are present as early as 6 months of age and pretty much maintained in comparison to both our low-risk group, our high-risk autism group, and our high-risk negative group. But contrast this to what we see in looking at amygdala development across the same interval in these groups. And here we see that in Fragile X, the amygdala volume is actually smaller, and the amygdala volume in autism is actually larger. Now, that's something that's been reported in older kids at 2 and 4 by our group and others. But the interesting thing here is the change over time. Again, we see no differences at 6 months of age, and these differences occur with an increased rate of growth of the amygdala between 6 and 12 months that levels off and is maintained across the second year of life. So here's a change in a structure tied to social behavior, social cognition, social development that's happening really as we're seeing the unfolding of social deficits in autism. And we haven't looked at that yet, but this is work by Mark Shen, and we're getting ready to do that. So where does this lead us? And it leads us to a discussion of how useful is this information in the public health arena. And so for a number of years, we have all tried to sort of peel the onion and find what are the behaviors early on that may predict or index later autism. And it hasn't been particularly successful, and partly for the reason, if not entirely for the reason, that the symptoms of autism are really just unfolding during this period of time, and they aren't really fully in place until 24 or 36 months. And so we have a parental sort of questionnaire called the First Year Inventory that looks at 12 months of age and was not able to predict autism at later ages, although they did predict developmental disability. And a paper that came out of the Babysit's Research Consortium by Kasia Jaworska that did a much more detailed analysis of 18-month data in a training set and a validation set, and found a positive predictive value of about 0.5, suggesting that they could identify sort of 50-50 the kids that developed autism. So this is at 18 months and predicting later autism at 36 months of age. So on the other hand, if we shift to thinking about early brain changes, as I've told you, we have a number of clear differences even as early as 6 months of age, and certainly in the first year. We see white matter tract differences. I showed you early on the fractional anisotropy, the inferior longitudinal fasciculus at 6 months of age. We see this hyperexpansion of surface area in the first year, increased size of the anterior corpus callosum, increased extra-axial CSF volume, and other things that we have from our study as well. And so this led us to start thinking about prediction. And I don't want to go into the details of the findings, except to say that we took data from the first year of life, brain imaging data, and we did it in several different ways. We did it with half of the set being a training set and the other half being a validation set, and we did it with cross-validation approaches of tenfold approaches. And the long and the short of it is that we were able to predict autism with a positive predictive value of 0.8 in 8 out of 10 children who were diagnosed with autism at 24 months of age. So again, correctly classifying 8 out of 10 infants. And I just want to add that this is a fairly conservative comparison. Let's see, I'm going to go ahead and check out what's on the slide. No, I'm going to go back. And that it's a conservative comparison for several reasons. The first being that we're comparing high-risk positive and high-risk negative. And the high-risk negative group, as we've seen in papers by Georgiadis and Messinger, is not a sort of pristine group that has no problems. The upper 25% or 30% go on to develop sort of subthreshold autism and language and learning problems, and now we know from other follow-ups other kind of psychiatric problems. And so it's a very mixed group. It's not like the typical approaches to these kinds of studies using machine learning that take a pristine sample of controls that have where everything's been excluded and a very clear sample of kids with autism. No one is sort of on the borderline, what we all tend to refer to as an elevator diagnosis. And separating out those kids is not quite the same as separating out these kids. Plus, we're separating these kids before the defining features of autism first appear. So this approach is not new. It's been well known in the degenerative brain disease literature that brain changes precede behavioral changes. One of the examples is in Parkinson's disease, where somewhere between 50 and 80% of the dopamine neurons in the substantia nigra are lost before people even develop symptoms of Parkinson's or come into the clinic. This is the same story with Alzheimer's disease and Huntington's disease. We see early brain changes and later behavioral changes. So this is really sort of the punchline of this talk, and I'm looking at the time. Maybe I'll end a little bit early. And I have no idea what happened to this slide. But the point is that this is where we have been for a long time in the autism field, where we cross a threshold in the community and kids are referred, and we diagnose autism, and we relate those to early environmental and genetic risk factors. But I think with these data and other data from a number of other researchers, we now see that there's this period of development where there's a lot going on, and the high-risk infant SID paradigm allows us to study this. In fact, I think there's so much going on that this is really the period of time when the proverbial horse is leaving the barn. And so I'd like to kind of compare this to the story in schizophrenia. I'm a child psychiatrist, and I might see a sixth grader who has a parent with schizophrenia and is not performing that well, and has learning problems and sort of schizoid personality features and ADHD, but they don't have the defining features of schizophrenia, which are auditory hallucinations and delusions. When those develop, and if those develop later on at 18 or 19, that's when we diagnose autism. And that's the common scenario in schizophrenia, that we see sort of prodromal features that evolve into full-blown schizophrenia. And I would offer that that's what we're seeing, at least in a lot of kids with autism, on a much shorter time scale. So we now know that there are lots of things going on during this interim period, sensory motor delays, atypical visual orienting, typical social behavior, and all sorts of changes in brain that develop with age-specific changes over time. So whether you call this a prodromal or a pre-symptomatic period, it precedes our ability to diagnose autism. It's a period of time when the brain and behavior changes of autism are being consolidated. And it's also a period of time where there's tremendous plasticity. And this is where we are, and this is where we've been in our efforts at early detection and early intervention. So we have to wait until autism declares itself in the community, and we try and push that needle down as early as we can with a variety of different screening tools. But I think that there's some reason to think, and we have, I think, some consensus in the field, although the data are limited, about early intervention being better. And we've seen the problems with later intervention in a number of examples, and I just cite one here as a pharmacologic trial of an mGlu-R5 antagonist in young adults with Fragile X. And this is a compound that worked very well in mice, and they went from having sort of misshapen dendritic spines to sort of pristine dendritic spines, and it altered behavior, and so it was tried in adults. But that, I think, misses the whole point that I think is the developmental perspective, that these adults
have had 20 or 25 or 30 years of living with a brain that's altered their experiences. They didn't go to soccer practice, and they maybe didn't go to their senior proms and drive, and those things all, as we know, shape and sculpt the brain. And so I think once we go through a period of development, there was a lot more complexity to this story than early on. And so that's why I make the case here that we should be doing or looking at early detection and early intervention in the same way that the degenerative brain disease fields are looking at in Alzheimer's and Parkinson's disease, to try and find biomarkers before the appearance of the disorder, which in some ways is, I don't think we would say in autism it's too late, but may not be the most effective time to intervene. So that brings me to this conversation about the environment and the psychological environment in particular. And I think since the sort of terrible stories that came out of the 40s and 50s and 60s about parental child rearing causing autism, we've all been a little uncomfortable thinking about the early psychological environment, but yet we know that even in typically developing children, parenting is important. And so here I add this to the model, not in any way to impugn parents or to suggest that this is a cause of autism, but just the idea that we can alter experience, we can alter experience-dependent neuronal development by our interactions, and perhaps have a more striking effect if we know the right things to do. And there's some beginning evidence that we may be starting to understand this. This is a paper by a group in the UK under the lead of Jonathan Green that was published last year using a parent-mediated intervention in high-risk infants and with a randomized trial with something called positive parenting, so sensitive parenting skills that were taught. And they saw some, although I think not the primary outcomes that were targeted, but some of the secondary outcomes that suggested that this was a very interesting approach and an important study. But I point out here that this is a hard study to do when you only expect 20% of the kids to end up with autism anyway. So you're really handicapped on the sample size, as opposed to having an early biomarker that might predict 8 out of 10 kids that would go on to develop autism. So this is another way that we're thinking about the environment, sort of a poor man's naturalistic way of looking at the language environment and the social language environment. A number of you, I think by now, are probably familiar with this wonderful Lena technology, the digital recorders that are sort of this memory stick size electronic recorder that fits into an infant's bib, and you can plug it into a computer and get a few measurements out of vocalizations, distinguishing the infant from the adults in the room, and things like back and forth or conversational turns. And what Megan Swanson found, that's currently under review, when she looked at this, is that she looked at low-risk kids, high-risk kids. None of these kids have gotten to the point where we would look to see if they have autism or not. But what was striking is this group in red, this group of high-risk kids that had a tremendously high rate of vocalizations, more than their low-risk peers, and of course this other group of high risk. So these differences are largely driven by this group. They don't seem to be related to conversational turns. And these infants engaged in less social babbling, as indexed by an item on the AOC. And so this is not what we expected. We expected that that there would be a subgroup that would actually be worse or lower, fewer vocalizations, kind of extrapolating from the idea of language delay being a common part of autism. But and Megan is interpreting this as more of in the domain of stereotyped repetitive behavior, non-social hypervocalization, and I think it underscores the importance of trying to augment and engage infants in social interaction, meaningful social interaction, because this is sort of working against us, if you will. So it's just sort of provocative preliminary data about a new idea that involves the sort of social language environment in autism. So I'm going to conclude, I think, with this statement, and that is it time for a pre-symptomatic clinical trial in high-risk infants with autism based on these findings of early biomarkers? I think we feel like that's perhaps a little premature, and we want to expand this and replicate this and have the rest of the field look for independent validation. But we think this moves us towards this idea of finding kids who are especially high risk for autism and intervening even earlier than we've had done before. So just to kind of conclude here with a few general observations from this kind of work that we've seen or taken away. And the first is that we see brain differences in autism present by 6 months of age during a prodromal period of development in autism. We see early brain changes that are largely in sensory motor regions, consistent with the clinical findings during the prodromal period. We see change over time in these developmental trajectories, such that it really is the change over time, if you will, that is the neural signature of autism, and not a static difference that's present. We see clearly age-specific brain changes. We have evidence that behavioral differences during this prodromal period can predict later autism and autistic behaviors, which I didn't go into. And finally, this idea, sort of a general conclusion, that early brain and behavior changes in autism may have or are likely to have downstream cascading effects in producing later manifestations of autism, as well as later brain changes. That's sort of, I think, linking back to Mark Johnson's interactive specialization. If any of you are familiar with that, I would highly recommend it. So this is our group that I wanted to acknowledge, the IBIS group, one of our annual meetings. We have folks at University of North Carolina. Heather Hazlett leads our local team. Martin Steiner in image processing. Hung Bin Gu is our statistician. Chad Chappell is our project coordinator. At University of Washington, there are a lot of people left off of this list, but Annette Estes and Steve Dager lead that group. WashU in St. Louis, under the direction of Kelly Badaron, with strong influences by John Constantino, Bob McKinstry, and John Pruitt, who really established our functional connectivity imaging program within this project. Folks at CHOP, under the direction of Bob Schultz, with Juhi Pandey and Ragini Verma. Lonnie Swagenbaum, who's been an instrumental part of this from Alberta. Guido Gehrig, who's now at New York University. And our data coordinating center under the direction of Alan Evans at McGill University. So I'm going to stop here, and I think we're going to move on with the program. I wanted to acknowledge the tremendous support that we've gotten from NICHD, Autism Speaks, and the Simons Foundation. And if you can imagine participating in a family like this, when you've just had a newborn and you have an older child with autism, and you often have to fly from where you live to where we are and spend a couple of days and come back 3 and now 4 times, that's a huge burden for families. And so we're very grateful to all the families that have made this made this study possible.
All right. Well, thank you so much, Dr. Piven, for a great presentation, and especially despite our technical difficulties. Now I'm going to hand the floor over to Dr. Megan Swanson, a member of the sessions working group, who will be moderating the Q&A session.
Thank you, Carolyn, and thank you, Dr. Piven, for this excellent talk. My name is Megan Swanson, and I'm a postdoc at UNC working with Dr. Piven. I'll be moderating the question and answer period today. Please, we're sharing a phone line here today, so please chat in if you can't hear either one of us. We have received several outstanding questions from the audience during the course of this presentation, and I'll start with the first question, but feel free to keep the questions rolling in. So, Dr. Piven, based on your findings, do you believe it's possible and advisable to diagnose ASD at 12 months or earlier, either in high-risk SIDS or in the general population?
Well, I don't think it's possible to make a diagnosis, certainly in most kids. There's a very nice paper by Sally Rogers that has an unusual sample because of her high visibility in the field, where she saw kids much younger than typical time points, and they looked very clearly autistic, and so she did an intervention study. I think what we're talking about is can we identify them pre-diagnosis with biomarkers? But I think the point here being that we actually can't make a diagnosis of autism in the first year, at 6 months or 12 months of age.
There's one that can't really see me very well. Oh, so, yeah. Okay, sorry about that. Okay, so in your opinion, what are the likely causes of the neural proliferation, which are likely the cause of these brain volume changes? Do you want me to repeat the question?
Yeah. I don't really know the answer to that. When it comes to the sort of basic biology, I'm really a lay person. We have evidence from the 16p11 studies that we see increased proliferation that has genetic origins, and we probably will find similar things in genetically defined autism, like CHD8 mutation, which is associated with big brains, and P10, which is another. I don't think we really know. I mean, it's obviously speculative to begin with to say that it's increased proliferation that's underlying this, so I wouldn't really presume to know what the causes of that increased proliferation would be.
All right. So among the brain measurements that you showed us today, what is the best predictor for autism or ASD during early development, in your opinion?
Well, predictor has been a term that's been used kind of loosely. We've been guilty of that as well. And it goes from one extreme of basically showing a correlation between an early brain finding and a later behavior to the other extreme, which is something that's clinically useful, where we need to look at sensitivity and specificity and positive predictive value. And so I think what we're finding is that there's no good evidence of a single structure or brain metric, and that we use more kind of complex approaches to model the complexities of the brain, and it's through that we're able to find later predictors of autism.
Okay. Are the imaging results bilateral or specific to one hemisphere?
Well, by and large, we haven't really looked at that. That's really the main answer. Clearly, there are some that are midline, like the cerebellum, the corpus callosum, and some that are lateralized, like the robust hyperexpansion in the middle occipital gyrus. But that's not to say that we aren't seeing differences in the left side as well. And so I think in full disclosure, we probably haven't adequately looked at laterality with our data yet.
Okay, I'm going to switch gears a little bit here. So do you think it's economically advisable to try to identify children in the prodromal phase, or more cost-effective to wait to see behavioral manifestations around the age of 2?
Well, I think what our data are showing us is that if you are at high familial risk, then it's certainly a plausible, viable approach that I think would have economic payoffs to do imaging in the first year. That said, eventually we would hope to have what we refer to in the field as surrogate biological markers that might map onto those imaging findings. We don't have them yet, but hopefully eventually it will be in the area of maybe electrophysiology or FNIRS imaging. But I think the caveat here is that this is in a group of kids that have already been identified to be at high risk. It clearly probably wouldn't work, and also is not financially feasible to do this sort of thing in the general population. But I think it's not unreasonable to think that with some of the advances in genetics, for example, with polygenic risk scores, that we might be able to sort of approximate the familial risk in the population with a simple saliva test. And if it becomes cheap enough, then we could use that as sort of a phase 1 screening that leads down the road to something of a more direct assessment of the brain with imaging.
Thanks, Joe. So on a related note, what are the ethical issues related to doing high-risk studies? So specifically, is there an ethical obligation to intervene or to notify families when early risk signs are identified, and to try to provide them with any sort of services or suggest services for them? How do you handle this in your study, in these longitudinal high-risk studies?
Well, this is a very complicated, understudied area for sure, and I think that it's very shortly going to become kind of front and center in our field. But I don't think we've really grappled with the ethical, legal, and social issues, the ELSI issues in this area. There was discussion of this at one of the earlier baby SIBS research consortiums, so I know that group is wrestling with it, but I think as a field, this is something that we're going to have to do. You know, with our group, I think it's always a balancing act. We don't want to provide information that's not replicated and clear, and so you know, it's all in the interpretation. So we can see early behavioral differences. Certainly, if we make a diagnosis of autism at 24 months, we make that known. But we've grappled with what we should do with our imaging findings at this point, because as we all know, in the field, there's a lot of non-reproducible results, and so we want replication before we can move this forward to something that's actually, you know, clinically useful. Okay.
Given that ASD can often be comorbid with other disorders, do you find any comorbid behavioral or neurodevelopmental disorders in infants during the follow-up period?
Well, there are a number of behavioral longitudinal papers now that show that those problems are present. In the high-risk negative group, the papers I mentioned by Georgiadis and Messinger excluded kids with autism and found that those kids at 36 months of age had significant problems, or at least in 25 to 30% of them. There are more recent papers by Peter Satmari's group that has followed these children into school age and shown cognitive deficits and functional deficits. And then, of course, we know that through family studies that eventually first-degree relatives are at higher risk for other psychiatric conditions. So there was a paper that came out this week or last week that I believe was profiled in the Simon's Spectrum newsletter that was a large Finnish sample that found high rates of psychiatric disorders in siblings in childhood. And so I think that this sort of increased genetic liability for autism is an important clue for us to begin understanding these endophenotypes and what are sort of necessary but not necessarily sufficient changes in brain and early behavior that go on to autism.
Okay. In a somewhat related question, how would you expect, oh, they just deleted my question. We'll ask a different question. Oh, there it is again. So how do you expect these findings to differ in de novo ASD cases when compared to familial ASD?
Right, well, that's a great question. I don't know the answer to it. I think that clearly we know, genetically speaking, that those kids are different, and we've done some family studies comparing single incidence and multiple incidence siblings as far as the phenomenology, their behavior, the broad autism phenotype, and we find differences there as well. I think right now it would be a difficult study to do because we aren't able to I think, do this study efficiently in the population. But eventually, if and when we can, I think that's a tremendously interesting question to find sort of other converging points that distinguish subsets of subgroups of people with autism in valid ways other than simply behavior. And so with the concordance of brain metrics, if we can converge on brain, behavior, and genetics, we validate these subtypes. So I answer to that question, I don't really know, but it's a very important question for us to pursue.
Okay. So how do we know whether the neurological events and brain changes are driven by heritable risk factors that are not initiated by the persons who later develop ASD, or these changes are due to some learning behaviors like imitation of parents or older siblings who have elevated ASD traits? Right.
Well, I seem to be answering all these questions the same way. I don't know the answer to that question. Having said that, I think that when the field sort of turns its attention in more rigorous ways to supporting these kinds of studies, I think we ought to be able to see differences. If we look at genetic risk markers, we might be able to see that those are more related versus a variety of environmental effects, both psychological and the sort of bioenvironment, and things like toxic exposures and air pollution may have a moderating effect as well. I think we are interested in looking at differences in parenting style, but really just as a way to get clues to what can be done differently, rather than as a way to sort of test out the idea of what's causing or underlying autism. But if we can create an environment similar to some of the early intervention studies, the Early Start Denver approach, that's really a kind of a sensitive back and forth interaction, and we have the right children targeted, then we might be able to really improve the outcome more dramatically early on.
Okay, are there candidate genes or signaling pathways that correspond to these ASD developmental brain findings?
Okay, so I'm going to trot out my same answer. These are tough questions, and they're too tough for me. We don't really know the answer to that, but clearly we need to integrate genetic studies looking at genes and from a sort of systems biology approach, gene pathways, and relate them to brain abnormalities. The problem with that is that we sort of live in two different worlds, and in the imaging world, we start with very small samples, and we make them even smaller because we're only studying 20% of those that go on to have autism. Whereas in the genetic studies, I think we've learned our lessons that at least in the common variants, common variation, we need very, very large studies. And so, I think we're going to have to find out some approaches that sort of cluster and consolidate the data so that we can begin to look at these, even if they're just sort of proof of principle kinds of findings. But ultimately, I think we will clearly find a relationship to more basic molecular and biology and genetics.
Okay, one last question, then we'll move on to some career development questions. So are ASD diagnoses at 24 months stable? Don't you think you should follow kids until they're 36 months of age or later before making a diagnosis?
Well, I think that that's an issue that we think about a lot, and we sort of frame it a little differently. Sometimes the implication from that question is that making a diagnosis of autism at 24 months in a kid who no longer meets criteria for autism at 36 months means that you were incorrect at 24 months. But I think that's a sort of a faulty assumption, and partly due to the fact that we don't know the natural history. And clearly the natural history trajectories is going to turn out to be that the trajectories are heterogeneous. And so there may be a subgroup of kids that look more like they have autism at 24 months and less like they have autism at 36 months. If I were a parent, and the sort of clinician said, well, the good news is that your kid may grow out of this at 36 months, I don't think I would find that too comforting. I think that what we have are children that meet criteria at 24 months, and we need to act on that. And some of those children will go on to have autism at 36 months. Some will be subthreshold for autism, and others will look pretty good. And so when we're trying to come up with early first-year predictors, it wouldn't make a lot of sense if you took that to its logical conclusion and we said, well, autism at age 5 or 6 is really much more stable. Let's do a study where we try and predict autism at 5 or 6. Well, really what we want is practical approaches to this is you predict autism at 24 months when it's first seen, and you intervene. And so I think there's a lot of confusion about this idea of stability and validity and what's the right way to do this. And I think we're really only at the beginning of that conversation. I don't think there is a right way. I think there are lots of different ways.
Thank you all for your excellent questions, and thank you, Dr. Piven, for your informative answers. Now, before we go, I would like to ask Dr. Piven about his career development and any advice he has for our early career researchers. So, Dr. Piven, can you start by telling us about your training trajectory, specifically what type of grad school you attended, if you did a postdoc, and any early funding that helped you launch your career?
Thank you, Megan. I think this is a hard thing to talk about to make it generalizable to the large number of people that are on this call. I think it really is different for different people, and my training going through medical school and psychiatry is a very different route than I think a lot of folks on this call who had did PhDs and graduate training. I think just in terms of the sort of influential things in my training, I finished, or I was coming to the end of my psychiatry training, and in full disclosure, I was headed out to private practice, and I was already interviewing at psychoanalytic institutes. So that's a little anecdote that may be interesting to relate to your own life. And when I was in residency, we had this opportunity to spend 6 months at the Institute of Psychiatry, and I just by chance wound up with Michael Rutter, who's probably well known to most of you, and as a sort of famous child and adolescent psychiatrist who really moved the field forward in dramatic ways. And that experience was kind of pivotal for me in getting me excited not only about child psychiatry, but about research. And when I returned to my residency, I had in mind a research project, and I decided I was going to try my hand at research. My goal was to do 1 year of research and use a chi-square and publish a paper. And I kind of got off that track when I had lunch with my soon-to-be mentor, Susan Polstein, who wrote the classic genetics paper with Mike Rutter, the twin study, when she said that, you know, if you're going to learn how to do research, you have to learn it from a mentor. And it was clear that I wasn't going to go anywhere on my own. And that she impressed upon me that it's important to take advantage of the opportunities in front of you. And I was going into this with a very fixed idea of the things that I wanted to study and I didn't want to study. And what she said was advice that I think I've used all throughout my career is when you land in a new place, you look for the opportunities that are strongest. And it might mean sort of changing your content area a little bit, but if that's the strongest mentoring and training experience, then you go in that direction, and then hopefully if you want to get back to your previous aspirations, you can, but more than likely you'll change direction. So then the other pivotal point was an experience with my division director in child and adolescent psychiatry at Johns Hopkins, Joe Coyle, who recognized my interest in trying to learn research and came to me one day. Now, this was ancient times, so it's hard to necessarily relate to today's world, but he asked me just for my CV. And so I gave him my CV, and unbeknownst to me, I think at the time, I can't really remember, maybe I'm embellishing, he submitted a grant to the John Merck Fund for one of the first John Merck fellowships, and came to me one day and said that he had 3 years of funding. He had some funding for me to use to buy equipment and to support a part-time research assistant. And I think that was really sort of the second point that I wanted to make that I think was very influential. That I quickly realized that that was probably the most satisfying part of his job. And it certainly has become the most satisfying part of my job, to try and sort of pass the baton and see if you can influence trainees in early development. I then spent time at the Johns Hopkins School of Public Health doing about half of a master's in epidemiology and biostatistics. So I moved beyond my chi-square, but just not very far, but in critical ways. And that was really necessary for me to be able to do research. Probably not an issue for most PhDs, but certainly for physicians, having training in epidemiology and sampling and biostatistics is very important. I moved after my postdoc fellowship to junior faculty position at the University of Iowa. What I found is that actually moves can be extremely refreshing and give you a new sense of direction, and that was exactly what happened at Iowa, where I found tremendous collaborators and was able to get a series of K awards, a KO1 and KO2, some private foundation funding, and small and R grant and R01s, and then moved to UNC about 15 years ago, where I got much more involved in research administration. So that's my path, and I'm going to turn it back to you.
So given your success at acquiring NIH dollars, what funding mechanisms would you recommend to advance young careers?
Well, that's kind of a tricky question, because I don't really know the answer to that, but there are certain principles, I think, to keep in mind. And one is persistence. I mean, we all know about probability and p-values, and if you push and you do it long enough, there are chances that things will get funded. And I've had experiences where things were helped over the line by a very benevolent program officer, and I think we've all seen this. And so sometimes strange things happen, but I think the key is persistence. I think another key in grants is, this may sound silly, but I think you obviously have to put your best foot forward, and what that requires is starting very early. So rather than being enticed into submitting grants much sooner than you should, it really requires, I think, months and months of discussion with mentors inside and of your institution and outside, and sending them aims and research plans to get feedback so that they can really shape it and vet it before you submit it. And then, of course, you know, there's this balance between having it be very solid, but also having an element of excitement. And I think that's where oftentimes collaborations come in, and cross-disciplinary collaborations, that you can add an element of excitement to what you're doing if you can figure out how to talk the language of another discipline and develop some meaningful collaborations.
So what foremost advice do you have for the early career scientists in our audience interested in following a similar path to academic success?
I'm trying to remember if this was on the list of questions, because, you know, I think, you know, the point I made about the kind of nodal points in my career were all about finding the right mentors. And at first it was an experience with Michael Rutter that inspired me, and then Joe Coyle in my child psychiatry training, Susan Folstein in my fellowship, and several other mentors after that. I think putting a lot of time and energy into finding the best mentor you can and the right fit is really kind of a critical stage. I think people that have that experience go on to be much more competitive in the job market, and people that don't struggle more. And so I think that that this is a tough environment, we all know, and I think choosing wisely your mentors is a critical step.
So, Dr. Piven, can you talk a little bit about some of the pros and cons of conducting research in an academic setting like UNC or possibly other settings?
Well, it's hard for me to talk about other settings, but I might sort of change the question just a little bit. That I think that there are a number of different pathways, you know, I've gone sort of a traditional route in academics. I've been in a medical school, and I think those of us working on the faculty in medical schools know that that's not always a very hospitable environment for research. You're sort of only as good as your last grant, and there's a lot of uncertainty. I've seen some of the junior folks from our lab go into sort of hard money positions in departments in psychology and elsewhere. I think that's a great route for a PhD if you can get it. It doesn't work so well for MDs. And no, I've completely forgotten the question. I'm going to pass it back to you.
Well, we'll wrap up. That was a great discussion. Thank you so much for the questions from the audience, and to Megan and Joe for a great discussion. And I just want to thank everybody who was involved with the session of the Summer Institute, Dr. Piven, of course, the working group, and everyone for joining us live. Video replays are available to INSAR members for the entire 6-course series, including this one. You can become an INSAR member by visiting autism-insar.org. If you have questions or want to get more involved, you can contact the Student and Trainee Committee at our email address, studentcommittee@insar-autism.org. And we hope that you've enjoyed this session of the 2016 INSAR Summer Institute. Please take a few moments to fill out the comment section after you end your session. We are eager for your feedback. Please join us next week when Connie Kasri will discuss parent and family-led interventions. Thank you.